Let's look at ecological interactions. How do pathogens penetrate plants? We've talked quite a bit about this as far as fungi goes. We're just going to repeat that part. Talked about nematodes. Nematodes were aggressive in the way they got in, because they have the ability to penetrate on their own. We talked about viruses and how viruses really need some sort of vector. We talked about bacteria, how bacteria are opportunistic. They need some sort of wound. They need some sort of hydrothode. Let's look at all of these things again. When we look at the disease cycle, we see this little chart. Plant growth begins. After that, you have what we call a pre-inoculation environment. We start getting to a point where pathogen is going to arrive. That's called the arrival of the inoculum. At that point, we have an obligate moisture period. Think of it like a seed. You put the seed and you water it. After you water it, it starts to grow. During that time, you're going to get germination. After the germination, you're going to get penetration of the material into the host cell itself. Then you start to develop the prospective disease. This shows you a spore that is germinated and it is now penetrating in. Then we get colonization. In colonization, we are talking about a post-moisture environment. Once the pathogen gets into the tissues, it no longer needs to worry about what the environment on the outside is, as long as it's not slowing down the pathogen and destroying it. We've got a post-moisture environment. During that time, we are going to get symptom development. You can see in this particular picture, you've got some symptoms developing on this tissue. Then we get sporulation. And when we get sporulation, that is going to cause the formation of inoculum that is going to be released, that will be dispersed. It will deposit itself back on susceptible living tissue, hopefully, and we start the process all over again. This is a disease cycle. What you have to understand is anytime you've got a disease that you're going to control, you have to be able to look at these parameters and figure out, how am I going to stop these parameters from doing what they're doing? We call all of this a disease cycle. The disease cycle takes in several different parts. In the beginning, there was going to be a germination process. That's where the spore lands. When the spore lands, it puts out these little germ tubes. When the spore comes in contact with a proper cell, it's going to form what's called an aprosorium. This is marked by this arrowhead up here. The aprosorium is where it's going to attach on. It forms an infection peg underneath of it. The infection peg pushes its way down through the cuticle and the cell wall into the cell below. One of two things are going to happen. It's either going to form a haustorium, or it's going to puncture that membrane and kill that cell. Haustorium, biotroph, puncture, kill, necrotroph. This shows you another one. You can see the spore over here on the right-hand part of the photograph. It's a larger mass. Then the little tube coming out, and then the aprosorium being formed over top, where it is penetrating through the host defenses of the cuticle and the cell wall. After it gets in, what we get is the formation of symptom. You can see the spore over on the right-hand side up midway towards the top. You can see the tissue over on the side, becoming discolored. Then we get colonization, and from that we get symptom formation. You can see the shiny little dark thing in there is actually the spore. The symptom around the outside of it, the size of this is about 1 millimeter. Then it's going to start taking over and really going out. When we were dealing with plant parasitic nematodes, remember we talked about the stylet. Stylet is basically an active mechanism. The organism takes that elongate stylet, comes up to the root, and pushes that stylet in to absorb nutrients. With the nematode, it is all active as far as the damage they cause. With viruses, remember we talked about transmission by insects and by other types of vectors. We talked about the non-persistent, the semi-persistent, the persistent type. When you're getting transmission from one crop to another, normally you're going to be looking more at the semi-persistent to the persistent type. We had different things. We talked about aphids, psyllids, winged aphids, whitefly, plant hoppers, nematodes, and midges. Then we talked about things like dodder. All of these are ways that the virus can be transmitted from one plant to another and as such cause damage. How do these things get into tissues, be it leaf tissue or elsewhere? One methodology is direct. We talked about that with fungi, where the fungus goes and penetrates directly. We have one group where they get in through the stomata. It doesn't matter whether it's a bacterium. Stomata are little openings on the outside of the plant. You can see a stomata right there and substomatal cavity underneath of it. When that's open, anything can migrate in. Over here, we've got a top view of stomata. You can see the stomata there. They can get in through the hydrathodes. We talked about the hydrathodes being the outer end of the vascular tissue. What happens is as the plant is nearing the end of the day, it's been pulling up moisture all day long. It stops needing all that pull-up of moisture. It takes it a while to shut down the pressure of pulling the moisture up. While it's doing that, moisture will actually go out the ends of the vascular tissue in what we call the hydathode. They will remain on the outside until something knocks it off. If nothing knocks it off, in the next day, these plants will often pull the material back in. If they pull that material back in and there's a bacterium sitting in that material, that bacterium is going to get in through the hydathode. If it gets in through the hydathode, then it's going to be able to transmit and start a problem there. Here is a leaf. You can see how this symptomology started at the junction of the vascular system with the outer margin of the leaf, which is called the hydathode. We have wounds for plant material to get in. This is sapsucker damage on a tree. What the sapsucker does is it comes around and it picks these little holes. This causes sap to come out, or nutrition in the phloem to come out. That will attract insects, and then the sapsucker will come back. It'll look for the insect, pick them out, and eat them. These serve as an area where something can get into the plant. If they get into the plant like that, they can be quite problematic.